Okay. So today we are going to be looking at the process of genome reproduction and genome maintenance on what we call genome expression. So that will be DNA replication and transcription. So for the first, you can see from this particular screen, a cartoon of the process that describes the whole idea of DNA replication. Remember in our last class, we were looking at the genetic materials, we looked at the DNA, we looked at the RNA, we looked at amino acids, and then we look at protein. And these are what we call the genetic materials. Now, these genetic materials are essential for the expression of the genetic traits that we're interested in studying. So when we talk about the DNA replication, what are we actually talking about? We are talking about the process that enables the reproduction of this particular genetic material, in this particular instance, DNA. Recall the fact that we have established some central dogma where we say DNA will give rise to RNA, RNA will give rise to polypeptide. And we also saw from our histories where we mentioned the fact that DNA molecule reproduces through what we call semi-conservative manner. And that was actually established somewhere in 1958 by Stoll and Messelson. So for this particular process to actually occur, as you can see here, the molecule must have what we call the origin of replication, or what is simply called the replicon. Without the origin of replication, this molecule cannot be able to reproduce. So this idea, as we have said, was proposed earlier on as the fact that the molecule actually reproduces in what is called semi-conservative manner. So three ideas were proposed: the conservative model, which was eventually rejected, but through the experiment of Michelson and Thor, they were able to establish the fact that this particular process actually occurs. Now, this is Thor, and then this is Matthew Michelson, Frank Thor, and these are the ideas that we were talking earlier on. Semi-conservative model describe a scenario where daughter cells will eventually have, daughter molecule will eventually have one whole of daughter molecule. And that is why you can see the parent molecule actually is purple in color. But by the time after the first few generation, you can see there is actually a blue and a purple color. So this is the semi-conservative fashion that we're talking about. And then in the second generation, you can also see what has happened. Now, in the conservative model, what that simply means is that a new set of molecules is reproduced entirely, which is not really correct. Now, if you go to the dispersed method, you can see some portion of a single molecule is actually made of blue and is also made of purple. So that also is not what is actually happening. But then let us come back to the semi-conservative model. Why do you think the semi-conservative model is actually viable? It's viable because it actually helps to maintain the integrity of the information in the DNA molecule. And so that is one thing that we must keep in our mind. And we have seen actually in one of our distance where we saw the elaborate experiment by Messerson and Storm on how they are able to come to the idea that this particular process is actually a semi-conservative. Now, let's see. Recall the fact that the bases in DNA, they are or nucleic acid, they are actually nitrogenous in nature. And that is why what they did was to actually grow this particular E. coli in two different culture. One culture having nitrogen 15, the other culture having nitrogen 14. And after a generation, what they did was to actually allow it to grow, like you can see from zero, from one to two to three to four generations, and then they actually centrifuge, and then they were able to actually study. Remember that 15 is actually a what? Is actually an isotopic form of nitrogen. So when they did that, by the time they centrifuge, they now discovered the fact that the quantity of nitrogen 15 is actually decreasing by half, by half over time. So that when they started with 100%, by the fourth generation, they had only about 12, which technically should be 12.5%. So through this particular experiment, they were able to actually establish the fact that DNA replication is actually a semi-conservative manner. So this is actually, you can take your time to actually look at that. Even the video, I'm not sure we are going to be able to look at or watch it, but at least.
In 1953, James Watson and Francis Crick built their model of the structure of DNA, the principal features of which are still accepted today. DNA consists of two strands that are complementary in base sequences to each other. A cytosine base in one strand always pairs with a guanine base in the other. An adenine base in one strand always pairs with a thymine base in the other. The complementary nature of the two strands suggested to the scientists a model for DNA replication. They proposed that the old strands serve as templates to make new complementary strands. The two resulting double helices would each contain one new and one old strand. There are other models to consider as well. For example, the old DNA molecule could be preserved and an entirely new DNA molecule could be produced from it. In yet another model, the result of replication would be two molecules with old and new DNA interspersed along each strand. From your knowledge of DNA replication, which one of these models is correct? Click on the correct model. From your knowledge of DNA replication, drag the correct names to the three models of DNA replication. Although Watson and Crick proposed the model of semi-conservative replication, at the time, no evidence existed to prove that this model was correct. To solve this problem, the scientists Matthew Meselson and Franklin Stahl designed an experiment to test Watson and Crick's model of replication. The key to the Meselson-Stahl experiment was devising a strategy to distinguish between old versus newly synthesized DNA. They distinguished the two by labeling them with isotopes. They grew Escherichia coli bacteria in the presence of either a heavy isotope of nitrogen, N15, or the ordinary light isotope, N14. After many generations, the DNA in the bacteria contained either the heavy or the light form of nitrogen, but not both. In this example, the nitrogen atoms in the thymine base are labeled with either the heavy or the light forms of nitrogen. The scientists took samples of each bacterial culture. They processed the bacteria to release the DNA into solution. Equal volumes of the DNA solutions were mixed together, and then this solution was mixed with a concentrated solution of the salt cesium chloride. The density of the cesium chloride was 1.71 grams per cubic centimeter, the same density as DNA. The tube was placed in an ultracentrifuge capable of high-speed centrifugation, 140,000 times the force of gravity for 20 hours. At this high speed, cesium ions had a tendency to sediment toward the bottom of the tube, forming a higher density solution at the bottom compared to the top. Other substances, such as the DNA, move to the position within the tube that matches their own density. Which band of DNA contains the heavy isotope, N15, of nitrogen? Click on the correct band. With their technique of separating heavy versus light DNA established, the scientists tested the hypothesis of semi-conservative replication. They first grew E. coli for 14 generations in a medium with N15 in the form of ammonium chloride as the sole nitrogen source. Growing the bacteria for many generations ensured that all the DNA would be labeled with heavy nitrogen. At this time, they isolated their first bacterial sample, prepared the DNA, and added the cesium chloride for centrifugation. At the same time, some of the bacteria were transferred to N14 light medium and allowed to continue to grow. From this point onward, newly replicated DNA will be made with the light form of nitrogen. They called their first sample Generation 0. After the transfer to light medium, a sample was taken every 20 minutes, which is the generation time for E. coli cells growing at their optimal temperature. The DNA from the samples was prepared for high-speed centrifugation. Messelson and Stahl found the following results. In Generation 0, all of the DNA was in the heavy form. After one generation, the DNA was neither heavy nor light, but an intermediate density. After 2 generations, half of the DNA was light and half was intermediate. After additional generations, more of the DNA was in the light form and less in the intermediate form. Generations 0 through 2 provide enough information to determine which model of DNA replication is correct. Click on the correct model. What would these hypothetical data suggest about DNA replication? Click on the appropriate model. Drag the appropriate density profiles to the generations on the left. Although we have examined what the data would look like for all 3 types of replication, keep in mind that Messelson and Stahl's real data match with semi-conservative replication.
All right, so we have seen the summary of the experiment conducted by Marcelson and Stoll in 1958. Now, I don't know when you have been able to pick up from this particular video, or quite a number of things. The scientific thinking behind the societal infrastructure on ground. As far back as 1958, they could actually maintain a scenario of 24 hours life supply. Now, this experiment was actually run for, the centrifugation alone was run for 20 hours. That's to say that you power your centrifuge and you leave it there for the next 20 hours. I don't know how that is going to be possible in this environment of today. And that's getting to almost 70 years anyway. Now, you recall also in 1958 precisely, here the molecule that was actually responsible for the reproduction of this particular molecule, DNA, was discovered by Atomberg and his group. Now, the DNA poly 3, which is called DNA polymerase 3, the anatomy is actually shown here for us. So the real polymerase in E. coli at least has 10 different subunits. They are the coenzyme that has 3 subunits, alpha, beta, and theta, epsilon and theta, and then alpha subunit is polymerase. Then the epsilon subunit is 3' exonuclease. The theta function is not yet known. The beta subunit dimer forms a ring around the DNA. Then enormous processivity, which is equal to like 5 million bases. So what does that supposed to mean? It means the capacity of the enzyme to actually add nucleotide, as is reproducing this particular molecule. So now you can actually see what is happening here. The various components are shown. You know, we have the chi, we have the sigma prime, and we have the gamma, we have the theta, the epsilon, the alpha, and the beta. All of them are here. So the core enzymes are alpha, theta, and epsilon. And then you can see the gamma complex on this other side, and you can see the clamp. The clamp is actually formed by the beta component. So what does it do? It actually holds the DNA molecule in place for the process or for the processivity to actually occur. Now, the features of this particular process known as DNA replication is actually one bidirectional. So when an origin of replication is identified, the process actually goes to the right and to the left. Then two, the double helix must be unwound by helicases. Remember when we're looking at the whole idea of the molecule, and we also saw that they are associated enzymes. Some of those enzymes we say they are replication enzyme, which we saw from our last class. One of those enzymes is called helicase. The other one is called gyrase or topoisomerase. And what does it do? It actually does the work of a supercoil must also be compensated. Then replication is semi-discontinuous. We'll come to see what that really means. And then the leading strand is formed continuously. Then the lagging strand is formed by Okazaki fragment discovered by Tuneko and Reggie Okazaki. That's where the Okazaki fragment is actually coming from. Also, this was discovered around 1968. So, DNA polymerase uses an RNA primer for that particular process to actually occur. Then a special primer forms the required primer, and then DNA polymerase will remove the primer at the end of the day. And then the nick or the space actually provided by the removal of this is filled by what we call DNA ligase. So if you look at it at the bottom here, it's a cartoon of the replication fork. You can see what we call the continuous or the leading strand, and then you can see on this other side what we call the discontinuous strand or the Okazaki. This is just one of it that we are going to see subsequently. And then you can see single strand binding proteins that are supposed to keep the molecule as single strand molecules. And then you can see the helices. They're supposed to do the unwinding of the helix. You can see the beta subunit that is holding the DNA molecule in place. Now, in E. coli, the replication consists of DNA unwinding proteins, the priming complex, which is actually called primosome, and then two equivalents of DNA, polymerase 3, that's the holoenzyme, which is very important, and then the initiation, the DNA protein bind to the repeats in the origin of replication to initiate this particular process suppression, and the helicases delivered by DNA-C further unwinds, and then primers then binds and construct the RNA primer. Remember, we said that for the process to actually start, we need an RNA primer. The RNA primer is actually formed by the RNA primase. So here, what we are seeing is a more larger replication for us to actually appreciate. You can appreciate the fact that remember that when you are reading in nucleic acid, whether RNA or DNA, you are supposed to read from the 5' to the 3', but that's the direction of movement. So in this particular instance, you can actually see that the 5', okay, the 3' is here with the blue, and the lower, then the 5' is one that is in the red and is going into the replication fork. But you see on this other side, the 5' is supposed to be coming from inside and going out. And because of that, it is a little difficult, and that is where the issue of the discontinuous fragment comes to be. So what happens actually in this instance is that small fragments of the RNA polymerase or the RNA primase will actually be put, as you are seeing here. The green color here is actually the RNA primer, and that is what you can also see here. So when they are actually laid, they actually continue to synthesize, and then by the time they get to the next RNA primer, the synthesis will stop, and then this molecule will be removed, and then the nick will now be sealed or will be replaced. This position will now be replaced by DNA, and then the nick will now be sealed by DNA polymerase. So now, remember that in this process, we have what we call initiation, then we'll have elongation, then we'll have termination. So the elongation actually involves DNA helicases unwinding single-stranded binding protein to keep the strands separated, as we have seen earlier on, and DNA polymerase grinding away on both sides. Then the termination, there is actually usually a position where that is called the tau locus, which is rich in G's and T's and is the signal of the end of replication. So a tau protein is also involved. Now, this protein is specialized in actually identifying this particular point, and is a contra helicase and prevents unwinding. Then topoisomerase 2, which is what we call DNA gyrase, it relieves the supercoiling that remains. So this is actually a cartoon for us to also appreciate the whole process. Now you can see the replication. The replication is a collection of all the proteins and all the other things that are involved in this process of replication. Remember, our leading strand will go into the fork, and our lagging strand, the synthesis of DNA or the process of replication will actually be coming from inside the fork outward. That is why you can have fragments, and these fragments are simply called Okazaki fragments. You can see here in the fragment whereby with this yellow portion actually standing for RNA primer, and then this other reddish portion is the DNA replication or DNA bases. They have been added until they will come, and then this portion will now be removed, and then this one too will now be removed, and then they will be replaced by DNA, and then the nick that this thing will now be sealed by DNA ligase. So this process is very important because the cell can never divide until the process of replication has occurred, and this is to ensure that there is enough and adequate amount of DNA has been reproduced to be shared between daughter cells. That is why we say like E. coli, but more complex. In humans, 6 billion base pairs are to be copied. Remember that we actually have 3 billion base pairs. 3 billion base pairs, but put together actually 6 billion bases. Now, multiple origins of replication, one part, 3 to 300 kb. That's to say, one origin of replication can be found within the range of 3,000 bases to 300,000 bases. Then several non-animal DNA polymerases, and then DNA polymerase alpha, 4 subunits, polymerase, the processivity 200, but no 3' exonuclease. Then DNA polymerase beta similar to alpha. So all this will actually come to work to ensure that this particular size of genome is actually replicated within the required time. And what you are seeing on this side is actually the cell cycle describing this particular idea. Usually, by the time the cell divides into daughter cells, they will now go through this cycle. The first is actually what we call the G1, and under G1 is rapid growth and metabolic activities. Then essentially is the S phase where this process of replication takes place, where you can see the actual level as DNA replication and growth. And then it will now move to G2, and in G2 you have growth and preparation for cell division, and then by the time it enters the M phase, then the cell or the mitosis phase, then the cell will actually divide into daughter cells. So the other thing we need to appreciate is the fact that DNA polymerase gamma, that is DNA replicating enzyme, is actually found in the mitochondria in the case of eukaryotic organisms. Then DNA polymerase delta has a 3' exonuclease as well as proliferating cell nuclear antigen. Then this proliferating cell nuclear antigen gives delta unlimited processivity and is homologous with prokaryotic polymerase 3. And the other thing we need to know is DNA polymerase epsilon is highly processive but does not have a subunit like the proliferating cell nuclear antigen. Now, another way to make DNA is actually reported in 1964 by Howard Taman, who noticed that DNA synthesis inhibitors prevent infection of cell in culture by RNA tumor viruses. So Taman predicts that DNA is actually an intermediate in RNA tumor virus replication. So that by 1970, Taman and Baltimore were able to actually report the fact that there is an enzyme that can actually reverse transcribe DNA. So how is that? They can actually pick up a messenger, an RNA molecule and reverse transcribe it back to DNA. And this is actually the basis for what is happening today when we group a whole class of viruses known as retroviruses, which HIV/AIDS is actually a classical case. Now, in the reverse transcriptions, you see primer is required, but a strange one. A transfer RNA molecule that the virus captures from the host. Then this reverse transcript transcribe the RNA molecule template into a complementary DNA to form the DNA-RNA hybrid. And all the RNA tumor virus contain a reverse transcriptase. So recall the fact that we need an RNA primer, primers to form our RNA primer. In this instance, what they do, instead of using that, they actually use the host machinery by commandeering the host tRNA and then use it to do this initial process of doing the forming the RNA primer, and then eventually converting it to C. Now, remember that cDNA actually means copy DNA or complementary DNA, and what that means is the fact that that DNA is a direct translation, reverse transcription from where? From messenger RNA. What is that supposed to mean? It means that it does not carry intronic sequences, that is sequences that do not code for amino acid. Remember, under normal circumstances, if you pick up a messenger RNA and you reverse transcribe, everything there is going to code for amino acid. So that's the difference between the normal DNA and the copy DNA. So 3 enzymes are very important in this process. Number 1, RNA-directed DNA polymerase. Number 2, RNA-search, and the activities is actually to degrade the hybrid that is formed by DNA and RNA. Remember that the DNA is actually a double-stranded molecule and is supposed to contain bases found in DNA. Then number 3, DNA-directed DNA polymerase, which makes the DNA duplex after the RNAs actually destroy the viral genome. And then the other thing we need to appreciate is the fact that HIV therapy, the AZT drug that is actually being administered, is what actually specifically inhibits this reverse transcription. So if we're able to stop this process from occurring, what it simply means is that we have actually halted the whole process of the virus getting into the genome of the host. And as a result, the disease could actually be stopped. So here you are seeing a cartoon that is labeled from 1 to 8. And number 1 is that at the simplest level is a double-stranded molecule like we have seen earlier on. Then number 2 is DNA is complex with histone to form nucleosome. We have seen that one in our last class. Number 3 is that each nucleosome consists of 8 histone proteins. That's why we say it has an octamer the octamer core of histone proteins. Number 4, nucleosome form bits. We saw that one even where we looked at the electron micrograph. Number 5, we also mentioned the fact that nucleosome fall up to 30 nanometer or 300 Armstrong. And then number 6, that forms loops averaging like 300 nanometer, which we also saw from our last class. Then number 7, we can see 300 nanometer fibers are compressed and folded to produce 200 and wider fibers. You can see we are getting to where? We're getting to 700 nanometer in diameter. And then finally, the number 8, which we saw that the tight coiling of the 250 nanometer fiber produces the chromatin of the chromosome. And everything put together, we actually have the diameter of the chromosome at 1400 nanometer. So remember, we have mentioned this several times that the only time we can see chromosome with the DNA with our eyes is in the form of chromosome, which is usually at the metaphase. So the process of DNA replication involves recognition of the position of DNA molecule. First of all, the idea of the origin of replication, because that is where the molecule reproduction will actually begin. Then there is elongation that concerns the event occurring at the replication fork, where the parent nucleotides are copied. Then when this process actually begins and is elongating, it has to stop somewhere. That is what we call determination, which is generally the only vaguely understood process among the three, when the parent molecule has been completely reproduced. So the complexity of this process: 1, separate the strand; 2, create primers. Remember RNA primers? 3, 5 to 3 prime leading strand, then 3 to 5 prime lagging strand. Remember we said there is a continuous strand and there is a discontinuous strand where we can see Okazaki fragments. And then the other thing we should remember is the fact that between Okazaki fragment or discontinuous fragment, there is a need to seal that, and we have an enzyme that does that is called DNA ligase, and that's why we say knitting together as a process that is called ligation. Then before we actually knit, we need to remove the primer, because the primer is actually RNA and is going to be containing uracil instead of thymine. And then the primer position is supposed to be filled with DNA, and that's what we call the primer replacement. And then finally, the ligation of these discontinuous fragments with the use of DNA ligase. Okay, maybe we should look at somewhere here. You can see this particular molecule is actually a circular DNA, and this is the point of what we call origin replication. Remember, replication is a bidirectional phenomenon. It goes, that's why the arrow is actually showing in the 2 directions. So this one is actually captured from electron microscopy, and a microgram is actually shown for you. This is a molecule that is actually in the process of being replicated. This is the actual, and then this is the drawing, the schematic diagram for you to actually appreciate. Remember that, or recall that in eukaryotic organism, the chromosomes or the DNA are usually linear. And that is what you are actually seeing here, DNA helix in a linear form, and then several original replications. We just saw that of man where we say in 3 billion base pairs. Now, these 3 billion base pairs, you find original replications scattered from between 3,000 to 300,000. So what does that supposed to mean? It means that in a 3 billion base pairs, let's assume that we are going to have um, we are going to have the 3,000 sequence or ranges. So in a 3 billion base pairs, what does that supposed to mean? It means that you're going to have thousands and hundreds of thousands of what of this particular origins of replication. And what is nature's idea, by the way, of having many replication origins is to be able to speed up the whole process so that the genome can be replicated within the shortest possible time. So this is the process that's actually been described from point number 1 to point number 4. You can see two templates needs to be actually separated. By the way, they are actually antiparallel, like we saw from our last class. Number 2, DNA synthesis will begin from 5 to 3 prime. Always remember that we must read DNA from 5 to 3 prime. Now, this one is actually coming out of the fork going to the outside, whereas this one is from outside going to the fork. So this is what we call the continuous, and this is going to be the discontinuous. Remember, fragments are going to be laid and stuff like that, and then this is the replication fork. And then the enzyme that is called helicase will continue to do its work of unwinding the helix so that the enzymes can have access to the bases to be replicated and stuff. So if you pick up that idea and you look into the molecular mechanism of what is actually happening, this is what you get to see. Now here we have the primer molecule that's actually been put. Remember that this primer is RNA primer. So it's been attached, and then subsequently the next one that is going to come is going to be a T, because this is actually an A, and the next one that is going to come is going to be a guanine, an adenine, because this is what? This is actually a thymine. So that is how the molecule is actually being replicated. So like we mentioned earlier on, the enzyme will include helicases, RNA primers, DNA polymerase delta, DNA polymerase epsilon, doing their respective work like you can see here. So this one is DNA polymerase, for DNA polymerase nucleotide polymerizing polymerizing enzyme first discovered in 1957 and reported actually in 1958 by Arthur Comberg. So this is the enzyme. The enzyme actually looks like a palm of your hand, you understand? And then you can see with the thumb that is actually trying to hold the DNA molecule. The fingers are actually seen on this side. Then this is the thumb, and then this is the palm. So that is the structure and the form, the morphology of the DNA polymerase. So other enzymes will include exonucleases, as DNA polymerase 1, for instance, that has the function of finding and removing RNA primases. Then RNA primers, sorry. Then DNA ligase that will add phosphate in the remaining gaps and seal it. And then nucleases removing wrong nucleotide from daughter strands. Now, these enzymes are there to ensure the integrity of the whole process. That the molecules, first of all, the bases are properly complementary. If there is an error, they need to be removed. The RNA primer is supposed to be removed at some point. The gap that is going to be created will now be replaced with DNA, and then the small nick that will eventually appear will now be sealed by DNA like this. So this table here depicts to us a number of the things, the enzymes. See the enzyme on this side, DNA polymerase 1, DNA polymerase 3, found in bacteria. You can see subunit 1, at least 10 in 3, like we have seen earlier on. The exonuclease activity, yes, for DNA pol 1, DNA pol 2, but the 5 to 3 prime is no for DNA pol 3. Then what actually happens? DNA repair and replication actually is conducted by DNA polymerase 1, but DNA polymerase 3 is a major enzyme for DNA replication in the prokaryotic system. That for the eukaryotic system, the DNA polymerases are listed alpha, gamma, epsilon, and delta. You can see also DNA polymerase kappa. The number of subunits are indicated. They are 3 to 5 prime, and then they are 5 prime to 3 prime direction, and what they can do is actually listed here. Remember that DNA polymerase gamma is actually the DNA that is found in, the DNA polymerase that is found in the mitochondria. Remember that the mitochondria has its own genome, and that genome also needs to be replicated. So that is why it has its own enzyme for that particular purpose. So this table again is a summary that you can take your time to look at the DNA polymerases in eukaryotic cells. Alpha, delta, epsilon, gamma, zeta, eta, theta, iota, kappa, lambda, mu, sigma, and stuff like that. 5 to 3 prime polymerase activity, you can see everything is yes. 3 to 5 prime activities, some are yes, some are no. So take your time to look at it. And then the cellular function they actually do. Let's take a few for instance. If we take the first one, alpha, we will see initiation of nuclear DNA synthesis and DNA repair as primase activity. If we take epsilon, for instance, leading, a leading strand synthesis, very important. If we take theta, you can see DNA repair. If we take mu and lambda, all of them DNA repair. And then if you take pi, you can see translation DNA synthesis. So where there are issues and they are removed, they need to be replaced. So this enzyme will do that work. Then components required for replication bacteria, they are also here: initiator protein, DNA helicase, gyrase, DNA primase, DNA polymerase 1, DNA ligase that will join Okazaki fragments and stuff like that. So you take some time to look at that. So in the process of DNA replication, DNA polymerase will require certain things. One, the DNTPs. The DNTPs are actually the bases, which we say DATP, DGTP, GCTP, PTTP, and then a cofactor known as magnesium, very important to stimulate and improve the overall performance. And then we need actually a primer chain with a free 3 prime OH, because recall the fact that that is the point of the reaction between the sugar and the phosphate. So then a template is required. The process of DNA replication will require a template because there must be an information that needs to be copied. That is, and this is nature's way of ensuring there is integrity, so that the system will not just start synthesizing something that does not exist. The integrity, let me use this analogy so that you can appreciate. Today in the human culture, we know we call people liars. And what do liars do? Liars tell you something that does not exist. You understand? Maybe you You say, okay, you heard of something like that actually existed in Abuja, Maitama. Then you are asking somebody who is coming from that place. Even though he may not be at the scene of the event, he can decide to start telling you what does not exist. So by the time you discover that what he told you was not actually what happened in the place, in your mind you can say, so this man was lying to me. So nature doesn't want that. Nature wanted that the integrity of the information must be kept, and that is why this process of genetic replication or DNA replication and genetic transmission of information is not compromised. It must be from the horse's, you know what we say, let us hear from the horse's mouth. And that is why DNA replication must have a template from where it's going to be replicated. Then five, double-stranded DNA that is fully intact and lacking a free OH will not be replicated. So we'll come to see that subsequently in the course, why this information that we just mentioned is very important. Above all, remember that DNA replication must be from 5 to 3 prime direction. When you are reading DNA, you are reading RNA, remember you must read them from 5 to 3 prime direction. All right, so the properties of DNA polymerase are actually shown on this slide. So you can take some time to look at it. DNA polymerase is a single peptide encoded by pol A. DNA polymerase 2 is a single peptide encoded by pol B. DNA has 3 polypeptide subunits. You know, they are listed there for you. Then DNA polymerase 4 is encoded by the B gene and is used in DNA repair. And then 5 or E, DNA polymerase 5 is encoded by UADC and is used in DNA repair. And one other thing we should also know is the fact that DNA polymerase unipolar is used for DNA replication is actually DNA pol 1 and DNA polymerase 3. Have 5 to 3 prime exonuclease for reading activity. Now, one of the things you must keep in your mind or you must remember is the fact that DNA is only the molecule in nature that has the capacity to actually correct itself. No other molecule in nature or in the living organism has the capacity to correct itself except the DNA. And nature has, because of the importance of the information DNA carries, that is why it has this capacity or capability to take care of itself if there are errors. All right. So you can see DNP plus DNTP is equal to DNP plus 1, and the pyrophosphate is actually released and the molecule is actually lengthened. That is why you can see N plus 1. So when you come down, for you to be able to appreciate the whole reaction is actually shown for you here. The reaction that actually occurs at the 3 prime OH. Remember, we said that is the point at which the reaction actually occurs. So what will happen is that if an incoming base comes in, it's going to actually react with the 3 prime with the phosphate. And then at the end of the day, 2 phosphate will actually be released in what is called pyrophosphate. And then if you come to this other side, recall from this particular side, you can see A and G. Now you can see the T has actually been added. And by the time the next molecule is going to come in, we now expect to see cytosine. Because on the template, we already have a guanine, and guanine will complement with cytosine. And so you can see, because there is actually a free OH, then there is the possibility that the guanine, the cytosine can actually be added. But if anything tampers with this particular OH, what that means is that there isn't going to be any extension of this particular molecule. So what are the properties of DNA polymerases from E. coli? Recall the fact that we say 5 to 3 prime polymerization and then proofreading in order to correct errors. We have the 3 prime to 5 prime exonuclease, and then also 3 to 5 prime, 5 to 3 prime exonuclease for error correction. And then in vitro elongation rate is actually 20 nucleotide per second in pole 1 and up to 1000 nucleotide per second in pole 3. So what does that supposed to mean? It means that the processivity in polymerase 3 is very high. Imagine adding 1000 nucleotide per second as against adding 20 nucleotide per second in polymerase 1. If you look at this again, you should appreciate the fact that when you write a letter, before you post it, or a message on your phone, before you post it, it is just ethical for you to go back and read to be sure that what you are about to post is actually what you intended the receiver to have. And if you read through and discover that there is a mistake, you try to edit. So this is actually what happens with this 3 to 5 prime exonuclease proofreading. After the molecules have been laid, replicated, what the enzyme will do is to actually come back and check to be sure that everything is okay. Then to proceed. Now, where there is an error, then the 5 to 3 prime exonuclease activity kicks in, and then it's able to take care of that. And who does that? Polymerase 1, as you can see. Who does the work of proofreading? Polymerase 1, 2, and 3, all of them do. Who does the work of polymerization? Polymerase 1, 2, and 3. And then the processivity recorded for polymerase 1 and for polymerase 3 is shown on the table. Now, protein recognition for origin of replication. Remember, we said DNA ABC proteins bind to the origin, then helicases will unwind, and then single strand binding protein will actually bind to ensure that the molecule has remained unwound. They don't actually collapse back on themselves. Then DNA gyrase and the topoisomerase will introduce negative supercoiling to give transitional strength. Now, remember the fact that apart from the fact that there is actually a helical winding of the molecule, and there is also an overall supercoiling. This overall supercoiling is handled by DNA gyrase, whereas the direct local helical portion is actually handled by helicases. And then RNA primers synthesize the primer as we have seen earlier on. And then polymerase 3 add the nucleotide to the primers. So this is what you are actually seeing here in this cartoon at the bottom. This is the origin of replication. That is where the process will actually begin. And when it begins, you can see RNA primer in green color on this other side going to the left in what is called the continuous or the leading strand. Then you can see this other one that is going to the right. You can see one fragment, two, and the third fragment. So these are the discontinuous, this is the discontinuous strand, or what we call the Okazaki fragment 1, 2, and 3. Remember that the RNA primase will eventually be removed and be replaced with DNA. And then the nick will now be sealed eventually by DNA ligase. That is again what you are also seeing here. You remember topoisomerase, DNA gyrase, you remember the helicase, local unwinding and overall unwinding. And then the single strand binding proteins, can you see them? Chromosome and so the whole of this here will now become the replication. Then you can see the RNA primer, that is being led by RNA primers. Then you continue, the reaction will continue. Remember, this reaction is coming out of the fork, whereas in the leading strand, the reaction is actually going into the fork. So the same thing here, protein machinery for DNA replication. So you take your time to get familiarized with it. Moving replication fork, DNA primers, RNA polymerase in the leading strand. So sometimes what we do in this particular course is that we can give you an image of a replication fork for you to identify some things or to label or to even identify what is actually happening in the picture and stuff like that. So again, this is another replication fork so that you can really appreciate what is happening. You can see on this side, you can see the largest strand coming from inside the fork going outward, and you can see from the leading strand coming from outside going inward. Enzyme unwinding the double helix and expose the bases. The replication fork is what you can see here, and then the free nucleotide, the DNA polymerase. So you take your time to get familiar with the story. Now, in this slide, what you are seeing is that you are seeing DNA extension and misincorporation, and then proofreading via exonuclease activity, and then DNA extension with correct base. Remember that it's a process, while nature has put in place all the mechanism to ensure there is error-free. The truth of the matter remains that there is no any perfect system in nature. So from time to time, there is actually the issue of incorporating the wrong base. And if the wrong base is incorporated, remember the DNA polymerase one, for instance, in the prokaryote organism will be responsible for the reading, and then eventual correction will take place. So they will be able to identify. So here, what you are seeing is that incorporation of incorrect base at 3' of the new leading strands, and then proofreading via mechanism. At some point, the exonuclease proofreading mechanism has identified, so it gets that removed. And then in the last diagram here, or the picture here, is that the correct base is brought and incorporated. So the error, but even with this, at the end of the day, there are scenarios where the wrong basic will still escape being identified and corrected. So let's keep that on. So that is how mutation will actually arise, number one. Number two, mutation will arise due to the fact that ionizing radiation and other chemicals and assorting agents will actually introduce modification that may lead to wrong reading. So the proofreading via exonuclease activity is actually shown here, just like we are seeing in the previous diagram. Incorporation of incorrect, G is actually complemented with T. Remember that G is supposed to complement with C, and T is supposed to complement with A. So in this instance, the enzyme, the exonuclease activity of the polymerase 1, for instance, is supposed to actually be able to detect the presence of this error and eventually submit it for correction. So these are all the ideas, so you can take your time to look at it. Now, model for the events occurring around a single replication fork. A, initiation, RNA primer made by DNA primers. Then B, order on twisting and elongation, new DNA strands, second Okazaki fragment elongation. Then here you have C, process continues, second Okazaki fragment, DNA primer, removed by DNA primers, polymerase 1, and then E, joining of adjacent DNA fragments by DNA ligase. Recall the fact that DNA ligase is usually the last enzyme to actually operate by sealing the final nickel. You also remember the fact that we say this whole process of DNA replication is required to take place before a cell actually divides. And Recall we just saw that in a cell cycle there are actually four phases. There's what we call the G1, there's what we call the G2, there's what we call the S, and then there's what we call the M, which is the mitosis phase. So to put it in the perspective, the S phase is actually the synthesis phase, which actually comes just before, which comes before the G2, and the G2 comes before the mitosis. Now, for G0 phase is actually seen in cells that actually enter rest, that they don't actually divide, e.g. the neurons largely are the ones that you can find in the G0 phase because they don't divide. And this cell cycle, even though we are not looking at it, it's just for a reference purpose. This cell cycle is tightly regulated because there are molecular, mechanism to ensure that before the cell can actually move from G1 to G2, all the checks have actually been done. By the time it enters the S phase, all the things that need to be done, the synthesis of all the DNA has been completed and everything will be checked. And then the cell is moved to G2. And in G2 largely is to make the completion to increase the size of the cell by way of mass and by way of increasing the organelles and everything in readiness for division. And by the time the cell enters the M phase, DNA material will condense into chromosomes, and then they will be aligned on the equator for movement and separation to the daughter cell through the process of cytokinesis. All right, so you can take your time to get familiar, like we said. So, determination of this process of DNA replication actually what? Pole 1 clips off RNA primers and fills in the gap, gold leading ligase strand, and then DNA ligase will seal the gap. So that reaction is actually shown in the cartoon below. Usually there is a small gap that has not been completed. That is what we call the nick. And you can see you bring in your nucleotide. Your nucleotide triphosphate will actually be added, and then this gap will now be sealed. So you can see eventually everything is sealed, and then AMP is actually released. Recall that ATP will come in, and the AMP will be released at the end of the day, and that particular portion is sealed. Okay. Let me say something here. DNA replication eukaryote, multiple origin replication, 50 nucleotide per second, autoradiography, and stuff like that. Now, these are some of the ways by which these ideas were actually established. They were established in a way that they did autoradiography, and they were able to actually see the fact that number one, there are several original replications. Number two, they are actually bidirectional in nature. And what did they use? They label with 3H thymidine for 10 minutes and add on label medium for 10 minutes to reduce level of new incorporated 3H thymidine. And then they were able to appreciate the process. So you can see the replication bubble is increasing and is going bidirectional. So the same idea is actually shown here. The four standard phases of eukaryotic cell cycle occurring, and replication occurring at the S phase. We have just seen that. Then different regions of the chromosome are replicated at different times. Arrow points to the replicating regions at different times. You can see, recall the fact that this is because of what? This is because of the fact that there are multiple origins of replication along the chromosome. So what are some of the things that we need to remind ourselves? Remember, number one, multiple replication origins occurring in clusters. 2. Replication unit activate at different times. 3. Within replication unit, replication region are separated by 330 to 300,000 pairs. Replication fork is replication forks form in pairs and create a replication bubble, which we have just passed. Then different origins on the same chromosome are replicated at different times in S. Then condensed chromatin replicates late, while less condensed chromatin replicates earlier. So here is actually just the demonstration. The search and identification of DNA replication sequence. So autonomously replicating sequence, only histidine expression can help to cells to survive. So you can see here, randomly selected yeast DNA segment and then segment of yeast DNA containing autonomously replicating sequence. So introduction of this plasmid to yeast cells that lack histidine gene and therefore cannot grow in the absence of histidine. Then selecting medium without histidine. So you can see the ones that are autonomously, they can replicate, but the ones that are regulated, you can see only two spots, whereas on this agar, you can see multiple. Also, rare transformers obtained. This contains plasmid DNA that has integrated into chromosome. And the high frequency of transformers obtained. This contains plasmid DNA cycle replicating free of the host chromosome. Now, this is what we call relaxed, whereas the other is called stringent, because here is tightly controlled, whereas on the other side is not. So the origin of DNA replication on chromosome 3 of the yeast Saccharomyces cerevisiae. If you look at it, you will appreciate this is chromosome 3. The telomeres of the chromosome are actually the ends of the chromosome, and then somewhere where the chromosome is actually connected is called the centromere. And then you can see all the green stripes. The green stripes are what? They are the origins of replication. So for instance, here, as shown by this cartoon, let's count and see 1, 2, 3, 4, 5. So there are about 9 origin of replication, just as shown on this for you to appreciate. Then a closer look at the order of replication in the yeast, you can see B1, B2, B3, other region binding required proteins. Origin of replication, then this order are very important for that particular process, because again, other region binding to required proteins for this particular place to operate. So it's just like you come to your house, you want to enter a house, there is a key or there is a padlock. How will you enter the house? You will need to have your key so that you can actually insert your key in the padlock and turn it. So that's what is actually happening here. While the original replication is there, you must also appreciate the fact that you need associated regions that proteins will be able to attach or bind to guide and for the identification of this origin of replication. So in humans, genes are more complex, even far distant DNA sequences can be important. So in a typical scenario here, you can see 50,000 nucleotide pairs and the origin of replication. So you can see the origin of replication here, and these are the what? DNA deletions in either of this region inactivate the indicated origin of replication. Now, what is the idea? The idea is that remember, they are supposed to serve as a guide. It's just like you having an address. Your uncle lives in Akwanga. Okay, you know your uncle lives in Akwanga, but you have never been to the house and you don't have the address. So you just stood up from Kaduna, you entered you went to the park, you entered your car and then land, eventually reach Akwanga. And then by the time you drop in a Akwanga motor park, you start asking people, I'm looking for my uncle, Mr. Moses Dauda. You say, Mr. Moses Dauda, okay. In which place, what does he do? You say, I don't know what he does. I don't know. You don't know what he does? Okay, where does he say he's actually living in Akwanga? I don't know. I just came because I just have to see him for this reason, for that reason. People will look at you as a stupid, because you don't have the necessary information to get to your uncle. And that is what actually happens here. These locations or these segments of this idea that we are seeing, they serve as a address, for the original replication. I remember that the audio replication is very important because the audio replication will actually allow for the reproduction of the DNA molecule. Otherwise, the molecule is not. So what does that supposed to mean? Otherwise, the cell will not grow. Simple. DNA proofreading usually doesn't have what? RNA usually doesn't have this. Yes, DNA is one. In fact, that is why nature deliberately removed the second OH group on the sugar so that it doesn't even introduce any anomaly. And that is why pairing correct nucleotide has high affinity binding to the moving polymerase. Paired nucleotide is easier to be off before covalent ligation. Exonucleotic proofreading and then strand-directed mismatch repair are all the things that can actually be done. So the proofreading sequence is actually shown here. Remember, we have just seen earlier on that if there is a mismatch, the enzyme will be able to identify. And when the enzyme identify, eventually the correction will be done by excising that particular nucleotide and then replacing it with the correct one. So we
Cells,
can
like
look at
these
this.
prokaryotic E. coli cells, replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
The other thing we need to know is how does this process actually occur? We have what we call the DNA repair process. One, excision repair. Just like the name implies, excision or lesion, new DNA synthesized, synthesis by pol 1 and ligase to seal the neg, which you have just seen. Actually, what is missing in this condition known as erythrodermal pigmentosis that is caused by a defect in the agent nucleus or any of the 8 other genes which removes pyrimidine dimers. What are the things that can assault the DNA? They are actually shown here. Cellular metabolism, ultraviolet light exposure, ionizing radiation, chemical exposure, and replication errors that will actually occur. All of them will lead to DNA damage. If there is DNA damage, then cell cycle checkpoint activation is expected to occur, transcription program activation, and then DNA repair, direct reversal-based excision, nucleotide excision repair, mismatch repair, double strand break, repairs and stuff like that. If at the end of the day all these are not able to take care of the situation, then the last option will come in, which is apoptosis. So the cell prepare to commit suicide. And the idea of this is to ensure that mutation is not actually transmitted to subsequent generation. But all the same, don't forget there are instances where this actually escape notice. So, mismatch repair is actually shown here, where you can see there is an X here, there is an A here. Naturally, it will create a bulge like this. Then DNA glycosylase will come and remove it, and after removing it, the endonuclease will then bring in the required nucleotide, and eventually you can see where an X was showing from the beginning, now it's replaced by a T, which is the normal complementary base. So that has actually taken care of that. Then there are what is called removal of uracil from the amination of cytosine. Sometimes all this kind of modification and structure could actually occur, but then nature has actually provided means by which this will actually be corrected. Then mismatch repair also in E. coli that we have just seen in the video can also be corrected. Now, DNA mutation rate is actually, by the way, one of the things that we must know is that mutation in DNA is a constant phenomenon. It's just like when they breathe air. It's just natural for you to breathe air. Now, when you breathe air, you actually survive. So DNA mutation is a natural and intrinsic property of the DNA. But it only becomes a problem when the mutation actually affects the coding sequence of the polypeptide. Otherwise, there is just a natural mutation occurring. That is why you can see here one mutation out of 100 million nucleotide per generation will actually occur. That is what is depicted here in this cartoon. Fibrinopeptides, hemoglobin, cytochrome C, then histone H4 5000 over millions of years since the emergence of species. The species are actually of the mammals from mammals, and then birds from reptiles, mammals from reptiles, cow from lamprey, vertebrate from insects, and so like that. So this cartoon shows us, nucleotide selection and DNA reading what actually occurs from 1 to 7. DNA polymerase pairs nucleotide during DNA replication. If incorrect, DNA polymerase is stalled until you remove the incorrect base and then replace it with the correct one. In 5, sometimes proofreading fails. We have just mentioned that the system is not that perfect, but efforts are actually being put in place to ensure that this does not exist. Then number 6, mismatch repair enzyme recognizes the deformity, and we have just seen that. And then in 7, replace the mismatch base with the correct one. So types of DNA repair synthesis systems in E. coli are listed in this table. Mismatch repair, base excision, nucleotide excision repair, direct repair. And if you look at it, under mismatch repair, you will see Dan-metallase, all the enzymes, DNA polymerase 3, exonuclease 1, exonuclease 7, exonuclease 10, DNA ligase, base excision repair, DNA glycosylases, which we have just seen. And what will they handle? They can handle mismatches, they can handle abnormal base, hypoxanthine, xanthine, alkylated bases, and stuff like that. Then API endonucleases, we have also just seen that DNA polymerase 1, we have seen that. Then under nucleotide excision repair, ABC exonuclear, there DNA lesions that cause large structural changes. And under direct repair, DNA proteolysis, pyrimidine dimers, then alkaline B protein. You can see 1 methyl guanine, 3 methyl cytosine. So these are all the kinds of damages they can actually correct. Now, remember or recall, like we have said, DNA is the only molecule that have the capacity to repair itself. Xeroderma pigmentosum is a classical case of the defect in DNA repair if the repair is not taking place. When people get exposed to ultraviolet radiation, like we have just seen constant challenge and assault by the sun, it will lead to this. By the way, let me put it this way so that you can appreciate for some of you that may not know. You see, skin color is actually adapted to the environment. So when you come to the north, you see people with dark pigmentation, especially in some regions of the north, where you get to see people voluntarily exposed to high sunlight. Color of their skin is actually darker compared to people who are in places where the sunlight and the ultraviolet radiation is actually low. So what is the idea? The idea is that this ultraviolet radiation will be absorbed by the pigmentation, thereby reducing the level of assault to the cell. So this is a classical scenario, because if the system is overwhelmed, what can you do? There's nothing much you can do. That is why nature actually added more advantage by reducing the level of assault, by increasing the skin color, so that this ultraviolet radiation can actually be absorbed by the darker skin. And people who are actually living in places that the ultraviolet radiation is low, they usually tend to have lighter skin color. This is to allow for this same ultraviolet radiation to be able to penetrate the skin for the process of vitamin D synthesis. So while the enzyme is important to do the work, that is under natural scenario. But then again, let me put it this way. If you have a small village that has a population of 200, they have their weapons to protect themselves. If that village is overtaken by 10,000 people, how can they survive? They will be completely overwhelmed and destroyed. So that is the idea. So while you have the DNA, you have the enzyme that will do the work of correcting errors and stuff like that, if you are exposed to high assault, definitely going to come down to contribute this scenario. So this is a classical expression of the xeroderma pigmentosum. As you can see, the skin actually been affected. Now, in the advanced world where they have understood the scenario and they have developed what to do, children and people who are suffering from this particular scenario will usually be given protective suit, because the simple science is understood that ultraviolet radiation reaching the skin is what actually causes this problem. So when they develop their suit, they actually give it to the people to wear. So you can see here different children and stuff like that, including these two adults who are actually twins. They were diagnosed at a very, very early stage, and they could survive into adulthood without any problem. All right, let's look at some inherited syndrome with defects in DNA repair. You can see the name of the syndrome is xeroderma pigmentosum. The one we have just seen is the most popular of them. But then beyond that, there are other ones: ataxia telangiectasia, BRCA2, which is a gene that is actually associated with breast cancer, ovarian cancer, prostate cancer. Werner syndrome, Bloom syndrome, Fanconi's anemia, and 46BR patients. To see what they are actually being affected, the phenotype, and then enzyme or process affected. You can see mismatch repair, nucleotide excision repair. You can see DNA interstrand cross-link repair, and then the last one here, 46BR patient, hypersensitivity to DNA damaging agents, genome instability, and affected DNA ligase 1. Okay, so these are some of the things that we need to appreciate, as we look at this particular scenario. So the summary of the whole story of DNA replication is actually shown on this cartoon. Number one, we should appreciate that helicases will unwind the parental double helix. Number two, single strand binding proteins will come and bind to the DNA so that they can remain open. Number three, leading strand is synthesized continuously in the 5 to 8 time direction. Number four, the largest strand is synthesized discontinuously from inside to outside. And then number five, after the RNA primer is replaced by DNA, then we actually have the final activity occurring, which is done by DNA ligase, and the full molecule is now produced, and then we now have the daughter cell. So these are the overall activity that actually occurs in DNA replication. So you can actually take some time to look at the conclusion. It is bidirectional, at least 3 different DNA polymerases, we have seen that. And then having replicate ends of linear DNA chromosome, and DNA at the ends of chromosome are telomeres. Then new histone proteins will be produced or be associated in eventual formation of our final molecule.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from reannealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA. The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
We have a lot of things to do, and we just have to do a lot of work. So the next thing we're going to look at quickly is what is called genome expression. In this instance, it's actually DNA transcription. And in DNA transcription, what we are just talking about the expression of the information contained in the DNA, and this expression actually done by way of synthesizing RNAs. So as you can see from this particular diagram, you have a eukaryotic cell with the nucleus, with the DNA that has been transcribed. Mature messenger RNA is produced, which eventually is transported to the nucleus, and then in the nucleus is being translated. So the process of actually copying information from DNA to RNA is simply known as transcription. And the resulting RNA, if course of protein, will be spliced and polyadenylated, transported to the cytoplasm, and by the process of translation will produce the desired protein. So that is what we just saw in that particular cartoon. Now, this is the process by which information coded in gene is converted into structures present and operating in the cell. By the way, don't forget, we mentioned the fact that proteins are the major machineries. The information, all the information contained in the genes will eventually end up in proteinous form. Now, these proteins will be the ones that will actually catalyze the synthesis and the incorporation of other macromolecules, whether they are lipid, whether they are carbohydrates. So the central dogma of molecular biology, DNA, we have just seen can replicate itself. We're also seeing now the information from DNA can be transcribed into RNA. RNA also will replicate itself, and then the RNA will eventually be translated into protein. And then there is a reverse transcription that can actually take place, which you can see from RNA to DNA. And also, DNA can bypass this process and go direct to translation into protein. So some of the things that we need to remember is the fact that we have our DNA. Proteins, keratin, collagen, for example, and stuff like that. And in the RNA, we saw also yesterday that there are a very large number of RNA types of RNAs. Okay. There are stages in this process of transcription, and they include what we call the transcription elongation, transcription termination, capping, and then polyadenylation. Remember, for us to appreciate this process, we need to look at the structure or the anatomy of the eukaryotic gene. The eukaryotic gene is usually formed by this structure, where you have the regulatory region, what we call the promoter region. Then you have what we call a portion known as the 5' untranslated messenger RNA region, and then the last or lower part or downstream that is called the 3' untranslated region of the messenger RNA, and then the poly-A, you can see it here. So, and within the gene, we have the coding and the non-coding. The non-coding sequences are the introns, whereas the coding sequences are the exons. And then there is what is called the start, and then there is what is called the termination. So these are things that we must keep in our mind so that we can appreciate. So transcription normally continues to a termination site, and in the case of the eukaryotic organelle, elongation requires transcription factors. This can be regulated by use of proteins or factors that we can see in Drosophila and other related species. So transcription stops. Polymerase and RNA are released from the DNA, and DNA rehybridizes in prokaryotes. Role depends on the sequence in messenger RNA which forms the stem loop. Now, one of the things we need to appreciate is the fact that when we look at the control region of the gene, there are quite a number of things. There is promoter, there is a target box. Target box is actually 32 upstream. Now, this blue color here and this arrow indicating going to the right is actually the beginning of this TK gene, for instance. So anything upstream here is actually labeled negative X. So minus 16, that's 16 days from the start of this particular gene. 32 meaning 32 days from, and then as you go upstream and stuff like that. So the promoter element within a typical eukaryotic gene are actually formed by what you can see here, based on the thymidine kinase gene. We have just seen that +1 means beginning of that particular gene. And you can see the tata box is actually -25, beginning -25. The cat and the GC is -50, -80, and so -130 and stuff like that. So elements of promote transcription are the +1 side, the tata box is located within this range upstream. Determine the exact start site, not in all promoters anyway. Then binds to the tata binding protein, which is the subunit of the transcription factor 2D. And elements that can promote transcription at other site, e.g. promoter proximal elements may contain the GC box. You see that we have just seen here. And then the card box, which is actually what we are seeing -80. And then the octamer that binds to the octamer transcription factor. So, and this, while this is essentially the same as you can see in other genes, there may be variation, or there are variation within the location as to the number of bases to the gene itself. Now, transcription factors are ubiquitous, and a few of them have actually been listed here, and they are listed here for you to actually appreciate what is happening. CREP, CTF, NF, you can see nuclear factor, then GALFO, galactose metabolism, then USF, upstream factor, GR, glucocorticoid receptor, or ER, estrogen receptor, TR, tyrosine, thyroid hormone receptor, MAC-C, a product of the C microoncogen, and then GCN4 amino acid synthesis. So these are not all, these are some of the transcription factors that are essential for transcription to actually take place, depending on the gene that is in question. The other thing we need to appreciate is the fact that there are classes of eukaryotic cellular elements. Now, ribosomal RNAs, like we saw in the last class, RNAs S, 18S, RNA 20S, 5.S, and 5, and then transfer RNAs, messenger RNAs, heterogeneous nuclear RNAs, and then small nuclear RNAs, which are listed as U1, U2, down to U10 and beyond, and then small cytoplasmic RNAs, and then what you see as 7SL RNA. So if you classify them, what do you get to see? The polymerase in the nucleus, for instance, RNA polymerase 1 will transcribe this RNAs. RNase polymerase II will transcribe heterogeneous nuclear RNA, messenger RNA, then small nuclear RNAs, and then U1, U2, U4, U5, and then RNA polymerase III will transcribe tRNA, 5sRNA, U6, snRNA, 7sRNA. And then for the mitochondria, remember the mitochondria is actually carrying its own genome and it needs to be transcribed. So RNA polymerase here, all mitochondrial RNAs will be produced by this particular RNA polymerase. Then eukaryotic RNA polymerase, you can see the error rate is actually in tens of thousands and it's highly processive. Then the sensitivity of the nuclear RNA polymerase to alpha amanitin is actually a toxin. So RNA polymerase 1 has resistance to that. RNA polymerase 2 highly sensitive, binds with this particular affinity, and then RNA polymerase low sensitivity binds with K is equal to 10 to the power of hundreds of thousands. Rho dependent requires a protein called Rho to bind to specific sequences. Now, one of the things you should appreciate is the fact that premature termination will actually arise, and this translation can be stalled by attenuation sequences. This one loops, which causes pausing, and then polymerase falls off. Can be regulated by tryptophan in tryptophan, and then anti-termination in lambda, for instance. The mechanism of termination is still being studied and being to be understood. For premature termination occurs in C, just like we have seen, and anti-termination mechanisms are known for HIV and TAP proteins. The other thing that can happen is the capping, which we have seen earlier on. We say there are actually 4 stages. Number 1, we say elongation of the process. Number 2, we talked about termination of the process, which we have just seen. Then number 3 is actually the capping. In this instance, the addition of 7-methyl-guanylate to the 5' of the pre-messenger RNA is the process of capping. Then it uses this 5' link. Reason involves instability, preventing degradation by 5' exonuclease enzyme. This is very important for us to appreciate. I don't know whether you will remember this 7-methyl-guanylyl acid is one of the modified RNA bases we saw in our last class. And I remember telling you that we are going to see the importance of this particular modified base. Recall again the structure of the eukaryotic gene. As you can see here, the control or regulatory region upstream. You can see the exon and the introns. You can see the poly A and you can see the termination. So remember we talked about 5' untranslated and then 3' untranslated region. At the end of the day, if this molecule is transcribed, you are going to have the exon and the introns produced as a primary transcript. Now, this primary transcript is supposed to undergo the next process that we call splicing, where the introns will actually be removed out, such that you now have what we call the mature transcript. Now, during that process, then the cap, which is formed by the 7-methyl-guanylic acid, can you see it here? This is the cap formed by the 7-methyl-guanylic acid, at the 5' position. The portion that is 5' untranslated, the portion that is 3' untranslated, and then the last process that occurs in the transcription process is actually the polyadenylation, which is the addition of poly A10, which is what you are actually seeing here in yellow. So introns, exons will be put together, and that's what you are seeing. And then when exons are put together, what you are seeing here is actually an example of what actually happens. You have your gene, you have your exon. So let's put this in perspective so that we can remember. The exons, which are the coding sequences, are usually a smaller part of the gene. So when you want to represent a gene by drawing, always keep in mind for you to give the correct idea that the genes are actually, the exon occupy a smaller portion of the entire gene. And so you can represent like 20% of the length can be the exon sequences, and the remaining 80 or more can be that is what you are actually seeing here. Now, when this is actually eventually transcribed, what do you get to see? You get to see. So now look at it. Look at the length of your gene from the beginning, and then look at the length of your gene after the transcript is actually produced. And this transcript will eventually be translated into the sequence of amino acids. So remember, we say polyadenylation is the last process that actually occurs. Then cleavage and polyadenylation specificity factor will actually appear to do the work. And then cleavage stimulatory factor will also appear. Then poly-A polymerase will add the poly-A tail. Now, if there's a process of copying information from DNA to RNA, remember we say we can produce all kinds of RNAs. But the most important one that we are much interested about is the coding sequence, the one that produces messenger RNA, and messenger RNA will eventually produce a protein or a polypeptide. Now, this process needs to be regulated. It is not just allowed to take place anyhow. That is why this segment of the class is called gene regulation. So the expression of most genes is controlled by the level of the transcription. These transcription factors will bind to promoter and enhancer to turn them on or off, as we have seen. Some of these are expressed in all cells, and at all times in what we call housekeeping genes. There are genes that are continuously being expressed, so they are active 24/7 because they enable the survival of the organism. But then there are also certain genes that will only operate at some point, and then they will be switched off at some point. And then some are expressed as cell enters a particular pathway of differentiation and during development and stuff like that. So this is a typical scenario of what actually happens. Remember, when the transcription factor aggregate, they must identify that gene that needs to be transcribed. And then they will come and bind to the promoter regions of that gene, and then eventually the molecule or that region of the DNA is actually transcribed. And this is what you are actually seeing here. The template is actually being used to transcribe the information. You now have your nascent RNA, which is the one in green color, while the reading continues in the 5' prime direction. So like we said, some of these are continually expressed, which we have labeled them as housekeeping genes. For instance, plasma cells express continuously the gene for antibodies, but for the antibody it synthesizes. And some are expressed only for conditions around and in the cell change. For example, the arrival of hormone may turn on or off certain genes in that particular cell. Recall that hormones are actually produced and make their action on distant targets. So when the hormones arrive, they are able to stimulate the cell, and the cell will actually respond appropriately. That's on the basis of the physiological situation. But the genes that are responsible for the respiratory function or the cellular respiration, they must be produced continuously for the cell to actually survive, to carry out certain, or to carry out the function they're supposed to actually carry out. Eukaryotes can respond to the environment. The main reason higher eukaryotes need to regulate their genes in cell specialization. That is why we have all kinds of different types of cells. The whole area of histology handles that. Then we look at simple tissue, we look at all kinds of tissue, we then go systematic. When we take the heart, for instance, we look at the different types of cells. Take the liver, we look at the different types of cells. We take the brain, we look at the different types of neurons that exist, and the glial cells, and all kinds of things and stuff like that. Anyway, this table shows all differences in gene expression between prokaryotes and eukaryotes. We need to appreciate the fact that in the prokaryote, all RNA species are synthesized by a single RNA polymerase. It doesn't matter the number of genes, whether 1,000 genes, whether 5,000 genes, one RNA polymerase will do that job. Whereas in the eukaryotic organism, 3 different RNAs, we have just seen it earlier in our transcription factors, are responsible for the different classes of RNAs. We saw it earlier. Messenger RNA is translated during transcription in the prokaryote. Now, the reason is very simple. The reason why number 2 point under prokaryote occurs is because in the prokaryotic organism, there is no nucleus. Whereas in the eukaryotic organism, there is nucleus. So RNA needs to be produced and be transported from the nucleus to the cytoplasm, where the process of protein synthesis occurs. Number 3, genes are contiguous segments of DNA that are collinear with the messenger RNA that is translated into protein. Whereas in the case of the eukaryotic organism, genes are often split. We just saw that a gene have 2 portions. The exonic portion, which is the portion that codes for amino acid, and the intronic portion, the portion that do not code. And then 4, messenger RNAs are often polycistronic, whereas in the case of messenger RNA in eukaryote, they are monocistronic. What is that supposed to mean, by the way? Polycistronic means if there are 3, 5 genes, all the 5 genes can actually be transcribed on one single RNA molecule, where you can have gene 1, gene 2, gene 3, gene 4, gene 5, and a single molecule. In the case of the eukaryotic organism, it is not done like that. Each gene stands independent. So that's why it's called monocistronic. We talked about the idea that there are genes that actually constituted So we said that some genes are ubiquitously expressed in what we call the constitutive expression. We also call them as housekeeping genes. Now this can be seen here. They are essential for the daily activities of the cell. Required for cell maintenance, such as actin tubulin, DNA polymerase, DNA, RNA polymerases. These are normal activities taking place within the cell. Then some are expressed in tissue-specific fashion. When you move from red blood cell to white blood cell to the beta cell of islets of Langerhans, to the hepatocyte in the liver, to the epithelial cell in the small intestine, or to the brain, to the neurons in the substantia nigra. They will give what they call tissue-specific expression. So here you have globin in red blood cells and then some expressed temporarily during development. Then the sum of the genes expressed in a particular cell determine the phenotype of that cell. From our histology, we know there are simple squamous epithelium. We have, and even there, when we move, we go to cuboidal, we go to columnar, whether ciliated, we go to cells in the liver, hepatocytes, that will have all kinds of shapes. We go to the pancreas, where we can see the acinine cells, we can see the endocrine cells, and even within the islets, we have different types of cells and issues like that. So for each cell, when you carry it, adipocyte, for instance, hepatocytes, whatever make them to look the way they are, whether by their morphology and by their function, is a collection of the genes that are being expressed in that particular cell. Another thing we need to know is that not all genes are expressed at the same time in a cell. Thus, there is a regulation of the expression of genes. Now, the other thing you need to appreciate is the fact that every cell has the total complement of the genes. If we say there are about or approximately 30,000 genes in the cell, in the human cell, what it means is that whether it's hepatocyte, whether it's adipocyte, whether is the epithelial cell that you find in the duodenum or in the stomach, in the esophagus, or whether it's the columnar cell you find in the stomach or in the duodenum, all of them, they have the same number of genes. What differentiates them is the genes that are actually expressed. So this is a classical case here. You have a liver cell here, and then you have lens cell. You see control elements upstream, promoter region. You can see promoter albumin crystalline gene. Now let's see what happens. Under A, you see albumin gene expressed because only activators, enhancers, and promoters are present for albumin. Are present for albumin present. So what does that supposed to mean? It means that the promoters and the rest, when they come, they will be able to activate this liver cell. Whereas for B, you have only activators for crystalline lens gene present in lens cell. So you can see here, only the ones here that can activate this. So albumin genes are not expressed, but that does not mean they are not there. They are there, but they are not expressed. The same thing here. Crystalline genes are there, but they are not activated because they are not being expressed. That is why the liver cell cannot produce the protein that are required for the lens, and the lens cannot produce the protein that are required to be in the red blood cell, for instance. Now, the idea of the gene regulation actually started with what we call the operand. And the operand is the combination of the operator and its associated structure, which we are going to see. The first and the most common operon that is understood is what is called the lag operon, or simply known as the lactose operon. Now, in the lactose operon, there are three enzymes. Number one, the panes that transport lactose across the plasma membrane. Two, beta-galactosidase, which hydrolyzes the lactose into glucose and galactose. Then three, the transacetylase, which function is still not understood. So these three genes are the associated genes. So in a typical operon model, this is what you get to see. The regulatory region is what you are seeing here upstream, and then what we call the OFR, which is simply the open reading frame. 1, 2, 3, they actually represent the genes or the structural genes that we have just seen. Then from here, what you are actually seeing is a promoter, and the RNA polymerase will come to bind the promoter region and begin the work of transcription. So recall we were talking about polycystonic messenger RNA. So by the time it transcribes this region, like Z, like Y, and like A, we actually have a single messenger RNA containing all the segments. So you can see message for like Z, message for like Y, and then message for like A. So this is what is called polycystone. So if you are given an image and you are shown that in that image there is the expression or message for gene A, gene C, gene Y in the same single molecule, and they ask you to identify what is it, then you know that you are dealing with a polycystone. Excuse me. So you now have your DNA, you have your like I gene regulation, you have the promoter region, then you have the operator, then you have the like Z, like Y, like A that is going to come subsequently. So what you have here is the messenger RNA, and then you have your DNA polymerase. Here you have your protein, which is the active repressor. So in the lactose is absent, the repressor active, and operon is actually off. So lactose is absent. So the gene cannot be active. If you don't have what to cook, will you go and on the cooker? The cooker, when you on it, is it just for fun, or is it for cooking? So that is the idea. So when there is no food to cook, then there is no need to go and on the cooker. So that's what actually happens here. If lactose is absent, the gene is suppressed. But if lactose is present, then the gene becomes active. So you can see here lactose present, glucose present, cyclic AMP level low, little lag messenger RNA is now synthesized. So what actually happens? The catabolite activating protein will bind to the promoter region. Now you can see an active cap is released. Then RNA polymerase less likely to bind, and then you have your inactive lag repressor is actually removed. So from here, you can then see that the operator of this particular gene is actually open for activity, so that this gene can actually be transcribed. Now, that is a classical scenario that you get to see in the prokaryotic organism. But in the case of eukaryotic organism, what actually happens is the fact that you have promoters, you have enhancers, you have silencers, and then you also have DNA methylation and hormones. Hormones is another way by which genes are actually regulated. So in a typical scenario is shown by this particular cartoon below. You have DNA and primary RNA transcript. The messenger RNA is produced, the messenger RNA is transported to the cytoplasm, Then within the cytoplasm, the messenger RNA is translated into protein. And then the other way to control this is to control protein activity, to make the protein inactive. That is just one aspect of the story. For the other ones, so many other things can actually happen. So if you look at this cartoon, then see what happens. Transcriptional control can actually, so onset of transcription can be regulated. Then translational control, where the protein itself needs to be produced, can also be controlled. And then post-translational control also exists, where protein activation by chemical modification, or you have feedback inhibition. Protein inhibits transcription of its own gene. Now, because we have so much, like we have the feedback mechanism that we get to see in endocrinology, we have enough protein, we don't need to produce more. So feedback mechanism will be sent back, and then the secretion of the hormone will be stopped. There are other things that can also help to regulate, and some of them are actually P-bodies, for instance, are classical things that can help to regulate genes, especially in the eukaryotic organism. Now, these are actually seen, associated with messenger RNAs in large aggregate called the P, or what is simply known as processing bodies. And the repression proteins actually break down messenger RNA by removing its cap, removing its tail, and degrading the remaining message in a small by small manner, which is called nibbling in 5 to 3 prime direction. The other one is what is called exosomes. Exosomes are actually large hollow macromolecular complexes that take up unfolded messenger RNA and degrade them in the 3 to 5 prime direction. And they are also important because they are actually associated with immune responses, viral pathogenicity, pregnancy, cardiovascular diseases, central. So many things associated. So as we are going to see, so this is a typical scenario of an exosome. Associated ideas. Now you can see fusion, receptor-mediated uptake, internalization. In the case of oligodendrocyte-derived exosome, it enhances internalization. In the case of fusion, exosomes derived or deriving from a platelet, melanoma, and glioblastoma cells, can actually do that. And then you can see tetraspanids, surface receptors, proteins, DNA fragment, messenger RNA, including microRNAs. They are all part of the things that you can see in the. Now, this exosome, they are an interesting group of macromolecular complexes because they are produced by cells and they are exchanged between cells. So now, what actually is happening here is that a group, now the overall function of an exosome will be determined by the origin of that particular cell. By that particular exosome, whether it's derived from cancer cell, whether it's derived from some other cell, and the function that they will perform will also determine what state. For instance, you saw in pregnancy, so many things actually happen in pregnancy. As the baby is actually, or as the fetus is actually growing. In the case of disease situation like cancer, so many things are actually happening, and that is why this particular cartoon here is showing us some of the hallmarks of exosomes. One, regulation of gene transcription and translation. Don't forget the major thing that we're talking about here. We're talking about how genes are actually regulated. We can see the exosome will do that job. Beyond that, they can do survival and proliferation, reproduction and development, angiogenesis and wound healing, host microbiome interaction and viral immunity. You can see apoptosis, receptor ligand signaling, cellular migration and metastatic disease, metabolic reprogramming, and regulation. So if you look at it like we have seen earlier on, you can see it's actually a small vesicle that actually contain so many things inside. And all kinds of things, all kinds of protein, amino acid metabolites, including our RNA, DNA, and microRNA. And we said earlier on that these microRNA are very important in regulation of this idea. So now this is the microRNA. MicroRNA, they themselves are actually small RNA molecules that bind to complementary portions of the U3' untranslated region of message and prevent it from being translated by ribosomes and or trigger its destruction. So remember that messenger RNAs are supposed to be single-stranded molecule. So, but then again, this small molecule will come and bind to this messenger RNA and make that portion double-stranded. So the moment other machineries will identify messenger RNA has been double-stranded, they will assume that, oh, this is a damage. And because it's a damage, it needs to be removed. And that is why they can be removed. So you can see here, protein coding region of target gene messenger RNA. So you can see the 3' untranslated region is free. So this will actually be what? Be translated. Now, on the contrary, when you come to this one, this microRNA have already bound to this 3' untranslated region. So this open reading frame cannot be translated, and that is why we are marking with X. So we have indirectly regulated the expression or the translation or the production of the polypeptide that this particular gene is supposed to. This idea has been extended, or like we have seen earlier on, in all kinds of applications. MicroRNAs regulate translation, and then this will also affect what we call synaptic structure and function in the nervous system. As you can see, microRNA dysregulation can affect microRNA population and can also increase alpha beta oligomers, tau hyperphosphorylation, TDP43 inclusion bodies, mitochondrial dysfunction, and then exocytosity. So the same thing, the synapse. Remember, we're talking about synaptic structure and function, which is very important in the nervous system. So if we go back to the gray box, which is what we can see here, all these development will lead to dementia. And this is actually as a result of the activity of microarray dysregulation. So how does microarray actually operate? Microarray operates like you can see here from the nucleus. You know, there is a microarray gene. The microarray gene will be produced into what is called the preform of it, and then the pre-microarray, then eventually it will be exported to the cytoplasm, and then in the cytoplasm, eventually the real microarray will be produced, and this will be complexed with proteins. This complex with proteins will actually eventually find its way. Remember that you also have your target. This is a target messenger RNA. Now, this target messenger RNA can actually undergo degradation by some other things, or can be suppressed and be repressed by this particular microRNA. So this is a video that actually describes the discovery of this particular phenomenon. It's a long video, but I think it's worth it to watch.
DNA might be called the blueprint of life, but RNA is the nitty-gritty instruction manual. RNA generally falls into two categories, coding and non-coding. Coding RNAs are messenger RNAs, which are transcribed from DNA and then translated into protein. Non-coding RNAs, however, have a variety of functions and make up over 80% of all the RNA transcripts in our body. In 2024, the Nobel Prize in Physiology and Medicine was awarded to Dr. Victor Ambros and Dr. Gary Ruvkun, the scientists who led the research that characterized a class of regulatory non-coding RNA called microRNA. In the 1990s, Dr. Ambros was studying the development of the nematode C. elegans. In particular, he was studying two mutant strains, one that was missing the Lin14 gene and one that was missing the Lin4 gene. The worms in both of these strains had developmental defects. The Lin14 gene encodes for a protein that controls the timing of developmental events. So it makes sense why worms missing this gene would have developmental defects. However, Dr. Ambrose was surprised to find that the Lin4 gene did not encode for a protein at all. Instead, it gave rise to a small single-stranded RNA that was only 22 nucleotides long. Dr. Ambrose confirmed his findings with Dr. Ruvkin, who was also studying the connection between Lin14 and Lin4. By comparing notes, they discovered that the small RNA derived from the Lin4 gene had a complementary sequence in the messenger RNA for Lin14. Because of this, they theorized that maybe the Lin4 small RNA acted as a post-transcriptional on-off switch that regulated gene expression of Lin14 at specific times during development. In normal wild-type worms, Lin14 would be expressed early on in development and then be turned off by the Lin4 small RNA at the correct time. Lin14 mutants would naturally exhibit the same developmental defects as a worm in which Lin14 protein expression was permanently off, since they are missing the gene entirely. But in Lin4 mutants, Lin14 expression would be permanently on, which also results in abnormal development. They published 2 back-to-back papers with evidence of a novel type of RNA that could regulate gene expression at the post-transcriptional level, rather than encode for a protein. A few years later, Dr. Ruvkin found another small non-coding RNA that also regulated development in C. elegans, called LET7. Just like Lin4, the gene for LET7 did not encode for a protein, but instead gave rise to a 21 nucleotide single strand of RNA that regulated the expression of many different genes that control development. What's more, LET7 is highly conserved and found in many species, from C. elegans to flies to humans. These discoveries cemented the existence of a class of non-coding regulatory RNAs called microRNA. Today, thousands of microRNAs have been reported and have been found to be involved in almost every biological process. MicroRNAs are being studied to treat genetic disorders, including cancer, autoimmune, and neurodegenerative diseases. They even have the potential to be used as biomarkers, an invaluable tool for early-stage disease diagnosis and preventative care. So a big thank you to Dr. Ambrose, Dr. Ruvkin, and their research teams for uncovering this small but mighty class of RNAs, and congratulations on winning the Nobel Prize.
Okay, that video is valuable as far as this talk is concerned, because so that we can really appreciate how scientific ideas are actually developed. You know, the another thing you need to appreciate is the fact that these guys have been doing this work for over 30 years, you know, trying to understand the mechanism for how microRNA actually works. And you can see from this short video how they systematically dissected and were able to identify microRNA as agents of gene regulation. And as you can see today, this idea is being applied to so many things, including diagnosis and treatment formula that has been developed for cancers and the rest. The other thing that actually helps to also regulate gene is actually what is called ribose switches. And in the case of ribose switches, it's actually the section of the 5 untranslated region. Recall the fact that the one we just finished now, which is microRNAs, they actually go to handle the 3' untranslated region. In the case of the ribose switches, they go to address the 5' untranslated region. And you can see here, remember, what you are trying to do is to control or regulate the expression of genes. And expression of genes can be done from whatever level. What you want is that you don't want the product, or you don't even want the gene to be expressed. And that is what you are getting to see here. Your ribose switch, you can see your ribose switch is actually coming and attaching to the 5' region So it's already on. If it comes to attach to the 5' region, it will actually disconnect so that the gene is actually not produced, is not actually expressed. Remember, this is your DNA. Your DNA polymerase have actually come to transcribe it, but then your ribose switch actually came on, attached to it, and by the time it attached to it, it was able to deactivate it, so that your RNA polymerase is there, your DNA molecule is here, and no messenger RNA is actually produced. The other one is that it will actually also operate at the level of protein synthesis. The first one is at the level of transcription. The second one is at the level of translation. You can see your protein synthesis is taking place when your ribose switch actually comes. It will attach to the machinery, and then it will turn off the machinery, so that your complex is actually disassembled and the protein is actually no longer produced. So that is what ribose switches will do. So the amino acid glycine and lysine, flavin, mononucleotide, and then S-adenosine methionine, the donates methyl group to many molecules, including DNA, and the cap of the 5' messenger will all be distorted. Okay. And in each case, the ribose switch regulates transcription of genes involved in metabolism of that particular molecule. And the metabolite binds to the growing messenger RNA and induce an allosteric change. And because of that, other gene enhance completion of synthesis of the messenger RNA. And in both cases, one result is to control the level of that metabolite. And your gene will no longer be expressed. Okay, eukaryotic gene regulation control points. Signal has arrived. When signal arrives, it actually is taken down through cascade of reaction into the nucleus. The gene is actually activated. You can see transcription occurring, eventual transportation of the messenger RNA into the cytoplasm. And in the cytoplasm, this messenger RNA will be translated into a chain of polypeptide. You can see here, and then this polypeptide can be active protein, and then the next thing it can do is to go transport to cellular destination, or it can actually be degraded. So whatever is the case, will be determined by the physiological state of the cell. If there is a need to pass through the pathway for degradation, it will actually go through the pathway for degradation. If it is okay, we still need supply of that particular protein, we will then continue to synthesize it and then target it to the destination. This is a classical instance, where you get to see your growth factor arriving to the cell and then binding to the receptor and then phosphorylation cascade, what we call signal transduction occurs, eventual transmission into the nucleus, activating the gene. The gene is actually expressed by production of messenger RNA. This messenger RNA eventually is carried into the cytoplasm for the process of protein synthesis. Now, the other one is what you see here. Testosterone, which is a lipid hormone, can come without this signal transduction event, will enter into the cytoplasm, will actually bind or complex with what we call a receptor hormone receptor complex, and then will find its way into the nucleus, activate the gene, and then the gene will be expressed. The messenger RNA will be taken to the cytoplasm, and then new protein will be produced. Now, for each of these proteins, they have actually what we call half-lives. And the proteins are actually listed, and messenger RNAs are actually listed here for you to appreciate. They have half-lives here, protein species, and then the longevity. If you take beta-actin, which is from non-muscle beta-actin, lifespan of the messenger RNA is 60 hours. and protein species is actually crystallizing. Life of organism 90 year plus. 6-glucose-6-phosphate dehydrogenase is 15 hours. It's actually hemoglobin. Maximum lifespan is 3 months. Insulin receptor, for instance, half-life is 9 hours. Seen in sesame milk, and then short-lived. Then you can see glucocorticoid receptor 2.3, and stuff like that. And then the last one here, ornithine decarboxylase, most bacterial messenger RNAs, less than 3 minutes. Remember that, the whole time it takes for the cell to even divide is 20 minutes. So that their own half-life is actually less than 3 minutes is not a surprise. So RAS cell cycle simulating pathway growth factor will come, and because it's actually a tyrosine kinase pathway, we can see what actually happens at number 2 tyrosine kinase receptor, and then dimerization through the process that leads to what we call signal transduction and eventual transmission of this information into the nucleus, and then the activation of the gene via expression of that particular gene, and then the messenger RNA coming into the cell, then protein that stimulates the cell cycle will be produced. So ideally, the proto-oncogene DNA, the proto-oncogene theory actually suggests that the proto-oncogene contained in the will be stimulated by many things. One of those is environmentally triggered scenario. When that actually occurs, translocation or transposition, a gene move to new location or locus under new control. And here you have normal growth stimulating protein in excess. Then there is gene amplification, multiple copies of the gene, normal growth stimulating protein in excess. And then the other one is point mutation within the control, oncogene, normal growth stimulating protein in excess. And then point mutation within the gene, oncogene, hyperactive or degradation resistant protein. So this is where you are going to have a lot of issues. So origins of oncogenes may have been viral elements, which for instance has actually been seen in Epstein-Barr virus, papillomavirus, and then HTLV1 retrovirus in what we call adult leukemia. Now, what was that supposed to mean? See, what it means is that there are genes that can predispose to development of cancer or tumors. And these genes may be there for all the time, and they may not be triggered. But then again, these genes can be there and can be environmentally triggered. And when we say environmentally triggered, we mean assault from the environment, which may include things that we ingest. That's to say, what we eat and drink can actually trigger that. Exposure to certain things can actually trigger that. And this also, classical example is Epstein-Barr virus. Epstein-Barr virus, a lot of people actually have carried the virus, but the virus, they will not do anything until it's triggered. Then it can precipitate the development of cancer. Now, the other regulatory pathway is what is called the p53 cell cycle inhibiting pathway. And you recall that we say so many things can initiate or can trigger environment, e.g. ultraviolet radiation can cause damage in the genome. And then these protein kinases, can trigger all sorts of things. Eventually, active form of p53 can actually be developed, which will go and actually suppress. It's supposed to suppress. And this suppression is what actually leads to apoptosis. So for here, you can see DNA protein that inhibits the cell cycle is actually produced. So mutation defective or missing transcription factors such as P53 cannot activate transcription. You understand? So as a result of that, you can actually have that particular scenario. So in essence, what we are saying is the fact that expression of information from DNA to RNA is simply called transcription. And transcription actually has 4 stages. Stage number 1, we say elongation. Number 2, we say termination. The process has to be terminated. Number 3, we need to add the cap. Number 4, we need to add the tail. And we have seen that this processes need to be controlled and regulated. And we saw several things that can do that. First of all, we saw P-bodies that can complex with messenger RNA and destroy it, removing the cap, removing the tail, and eventually eating of the molecule. We saw microRNAs, we saw exosomes, and we say exosomes are also there. They help in the regulation of genes and stuff like that. And we saw that they are actually exchanged between cells, and whatever they are able to do is actually determined by their origin. Then number 3, we saw microRNAs, and we say microRNAs help to regulate gene expression by way of actually attaching to the 3' untranslated region, thereby making the molecule completely suppressed. And then the last one we saw ribose switches. Ribose switches actually go to the 5' untranslated region, and they can control at the level of transcription, and they can also control at the level of translation. And we also saw quite a number of other things that can actually happen. The different levels of regulatory control that can occur in eukaryotic system. So we need to appreciate that. By the way, let me also say this. You see what we have done from last class, which is when we look at the genetic molecules, and what we are doing now, looking at the maintenance and expression of the genome. Next class, we will complete the remaining thing, which is actually transporting synthesis. These are the things, the core science, in genetics that every one of you must take time to understand. Because subsequently in the course, what we are going to be doing is to be applying this basic knowledge, to some of the things that we are going to encounter. And we'll take some time to look at transcription and transcriptomics. We'll take some time to look at proteomics, and we'll describe some of the tools, some of the techniques. You understand? So these are some of the things that you need to know from now. So we can stop this class now. And if there's any question that somebody want to ask, you can actually ask before we close. If there is no any question, we will continue. We may continue tomorrow or on Friday. So I will need to look into my timetable so that I can get the appropriate date. Like I said, we still have a lot of things to do, and we need to actually double up our classes, because I was told that exam is coming and stuff like that. The other thing I will also need to know from you people, what happened to the tests that we say we are going to write. So you can put your comments on the WhatsApp platform. Thank you very much.